Hello guys, welcome back in another video tutorial for microbiology with Shagnik. So in that video tutorial, I am going to discuss with you about the concept of melting temperature or TM of the DNA. So let's get started with this video. So the melting temperature or TM of the DNA is the temperature at which 50% of the DNA sample is melted or denatured. Okay, so what is the idea? The idea is, suppose this is a DNA molecule, and remember, in between the DNA strands, there will be hydrogen bonds, right? So the idea is melting temperature is the temperature at which half of the hydrogen bond in that particular DNA molecule is broken down. Okay, because remember, temperature can facilitate the denaturation of the DNA. And how this denaturation will going on? The denaturation will going to occur by breaking of the hydrogen bond, right? So that is the idea. So the temperature at which half of the hydrogen bond in that particular DNA molecule will be broken, we will call that particular temperature as Tm. Okay. So melting temperature, remember, is especially dependent on the GC content of the DNA. Okay. Remember, the Tm value depends on GC content of the DNA. Now why this so? It is because, remember, in between G and C there is 3 hydrogen bond, whereas in between A and T there is 2 hydrogen bond according to Watson-Crick base pairing, right? And remember one thing, that we need more amount of temperature for breaking 3 hydrogen bond than AT. Okay, that is why the amount of GC content in a particular DNA molecule will going to determine the amount of temperature required to break them. Okay, that is why, remember, the higher the GC content, the higher will be the Tm value. Okay, so we will going to see two graphs for understanding the concept of melting temperature very well. So this is the first and the very basic graph, that is, here we have taken absorbance value at 260 nanometer in the y-axis and temperature at x-axis. Okay, now remember one thing, that is, we usually measure DNA at 260 nanometer, whereas we measure protein at 280 nanometer. Okay, that is very important thing, and I usually confused between them often. That's why I have told you. So here we will going to get graph of something like that. Okay, as you can see, the DNA is denaturing with increase in the temperature, right? And this is the point which we will call as melting temperature, where the DNA is partially unwind. Partially means 50% of the hydrogen bond is broken down. Okay, so, and then when we will going to increase the temperature, they will be totally converted into single stranded DNA. Okay, so now I am going to show you a experiment that you may probably have done in your university or in your college during your BSc or MSc times. So let's see. So this is the instrument that we use for the practical of melting temperature or TM. So here in this container we have DNA sample, okay, that we have prepared, and this is a glass rod to swirl them. Okay, so the idea is, initially, when we have not, and before going to explain it, I want to told you the use of this instrument is, this instrument will going to provide heat to that sample. Okay, so now, initially, when we have not started this instrument, we will going to take some amount of sample in the cuvette. Okay, and we will going to put this cuvette in spectrophotometer. Okay, and what we will get, we will get at 260 nanometer a OD of 1. Okay, so now after that, with time, when we will going to take sample again, remember we have to take sample after every 5 minutes. Okay, so with time, when we will going to take samples, we will going to see that the OD is constantly increasing. Why is this so? because as DNA is denaturing, so here I am drawing DNA in a linear portion so that you can understand it well. So as DNA is denaturing, remember the surface area is increasing, right? So more amount of light get the chance to be absorbed, right? Because the surface area is increasing. Because try to understand, initially we have DNA in such conformation, right? So here light only can be absorbed by this portion, this portion, this portion, this portion, then here, here, here. But there is no absorbance here. But as the DNA is denaturation, the light can also absorb by this part of the DNA, right? That is why with time the OD will going to increase. But here the surprising thing is, after certain point the OD will going to be remain constant. Okay, and the OD value where the or after which the OD are not increasing, remember that particular OD value is the value where we will going to get the TM after plotting that OD values in that graph we have just discussed. So this is the graph that we have got when we have performed that experiment. So that is the idea. Okay. So now with the help of that OD value, we can easily find out the temperature by extrapolation, right? And that particular temperature will be our melting temperature. Understood? So that is the idea. Now see, here there is another graph, and this graph will going to explain why I have told you that GC content will going to play a very important role in the determination of TM. Here you can see when the GC content is just 35%, at a very low temperature we have got that TM value, right? But when the GC content is 50%, we need a more temperature to get the TM value, right? Or the temperature of the TM value is more. And when we have used a DNA of 66% GC content, we have seen that the TM value is at higher temperature, right? So that is the idea. That simply tells us that the higher the GC content, the higher will be the melting temperature, right? Now I have here written various type of factors that can affect TM value. So first we have already discussed that the GC content, the higher the GC content, the higher will be the TM value. Now remember another thing, that in increasing amount of Na plus concentration, TM value will be lower. Okay, because remember one thing, just a minute. We all know that the sugar phosphate backbone is present in DNA, right? And this phosphate have negative charge, right? I hope it is clear to you. Now, what Na plus concentration will do, Na plus will going to shield those negative charge. Now, you may ask, what will going to be the consequences of shielding those kind of negative charge? Now, remember that when the backbone of the DNA has many phosphate group unshielded, this phosphate can interact with each other. Okay, and this interaction creates helix destabilization. Okay, that means this kind of interaction is not good for the DNA, right? Now try to think about this. If DNA itself promotes its destabilization, then it will be easier for the temperature, or I can say that at very low temperature the DNA can be denatured, right? So that is the thing. That means in absence of Na plus concentration, TM value of a particular DNA will be very low, right? Because helix destabilization due to interaction between negatively charged phosphate already supporting the destabilization or the denaturation of the DNA, right? But when Na plus is present, what happens? Na plus will going to shield those phosphate backbones, right? And as a result, they cannot interact with each other. And as a result, DNA becomes stable, right? So the entire thing for denaturation of the DNA, now TM have to manage, right? So that is the thing why in presence of Na plus concentration, DNA having same sequence have higher Tm value, and in absence of Na plus, the same DNA have a lower Tm value. Understood? So here there is an important equation that can tell us about the relationship between GC content and the Tm, that is percentage of GC is equal to Tm minus 69.33 into 2.44. Okay, this is the equation that help us to understand the relation between the melting temperature and the GC content. Now here another important formula, that is the zipper effect formula, applicable for short DNA nucleotide of 25 to 30 nucleotide DNA, that is Tm is equal to 2.2 degrees centigrade into A plus T plus 4 degrees centigrade into G plus C. Remember, this formula will going to be very important in our recombinant DNA technology, because when we will going to design PCR primers, this value will going to help us to determine the Tm value. Okay, because determination of the Tm value of that primer is very important for the creation of that particular primer. Remember it. Okay, and we will going to talk about this in details when we will going to do a lecture series on PCR. Okay, so that is the idea of our Tm value. I hope it is now very clear to you, right? So here you have learned that GC content will going to positively affect the Tm value. That means higher the GC content, higher will be the Tm value. In presence of Na plus concentration, the Tm value will be higher, right? That is the two factors. Okay, along with the length of the hybrid DNA will also going to affect the Tm, because higher the length, higher will be the hydrogen bonds present, higher will be the temperature required, right? So Tm value also increasing, right? And here the main important thing for your university exam is the definition and that particular graph. And for any kind of entrance exam, the entire thing is important, right? So that is the video for our melting temperature of the DNA. I hope it will be helpful for you. Thank you.